So before we go for the next plenary session, we have a small announcement to make. So I just call Dr. Robin to come for the announcement.
Hi everybody, I'm Robin from ISER Tirupati, and I think as you would have noticed, there are a lot of people who are beginning to start working with bioacoustics. It's a relatively new field, and some of us are kind of trying to get together to create a center, bioacoustic center in India, and the goal of the center is to increase training so that people can do more effective work with a tool that is definitely going to be very popular. Because unlike genetics and many other tools, bioacoustics is very easily accessible and it's very applicable for conservation. So if you're interested in bioacoustics, if you do any research on bioacoustics, we will also have training programs and things like that, workshops. Please feel free to join us. This link will take you to a Google form. At the moment, there are mostly academics, people who are interested in research and wildlife conservation, but we also have other folks who are interested in applied aspects like understanding conflict. We have civil society organizations who are trying to use acoustics for such uses. There are also companies that want to use acoustics for measuring biodiversity. So this, I think, is going to be a very exciting new field that people can kind of participate in. So please, if you're interested, feel free to join in. Thanks.
Good evening. I would like to introduce our next plenary speaker, Mr. Ramesh Kumar Pandey, who will be talking about the power of partnership, linking research and policy for lasting conservation impacts. Mr. Ramesh Kumar Pandey is the Additional Director General of Forests, MOEFCC, who is an Indian Forest Service officer with nearly 30 years of experience and has distinguished himself in wildlife protection and enforcement. While he was the field director of Dudhwa Tiger Reserve, he implemented the M-Stripes patrolling system and led enforcement operations against tiger poachers and wildlife traffickers. In recognition of his outstanding transboundary environmental crime record efforts, he was awarded the UNEP Asia Environmental Enforcement Award in 2019. Over to you, sir.
Good afternoon, everyone. First of all, I would like to thank Director, Wildlife Institute of India, and Dr. Bilal Habib in particular, because he was consistently pursuing to invite and willing that I should speak on how the research and policy can be bridged and the evidences which are coming through research can be translated into action. I don't have any structured presentation. My team had tried to make one, but I didn't get time to go through it. So kindly bear with it, and will be extempore, and whatever comes to mind will be shared with you. And you may agree, disagree what I would like to profess. So when I personally see that how the conservation or wildlife statutes or regulatory provisions or policies have taken shape in India, I feel that it came from top, I mean from whether it is provisions in the Constitution. Suppose we start from 1950. So you might have heard that there are provisions in Constitution for the protection and conservation of wildlife. In those days also, people were thinking of having a provision in Constitution to protect wildlife. It got mentioned in directive principles to the state that every state has to protect the environment. It also came into the fundamental rights. And that time there was, people had anecdotal stories, people had all kinds of books written about the games and all kinds of games, animals and enjoyment they derived in games. And then a time came around 70s when everyone suddenly started talking about environmental conservation. And in India also, people started thinking that we should have a statute in place, and a Rio conference happened. Then the Wildlife Protection Act came in 1972. Project Tiger happened to start in 1973. CITES, the convention which deals the endangered species of flora and fauna, came into 1973, and you will be surprised to know that the first convention or the second convention of the CITES happened in India. So these things were happening in early 70s, and we didn't have any scientific temperament for the wildlife, and wildlife mostly was a subject confined to rich families, rajas, jamindars who used to go jungle and could afford camping in forest and taking hunting permits. So even then, we didn't have any concept of having research kind of thing and trying to take the inferences of research into the policy. I personally feel that after that, a phase came when people started observing wildlife by rearing them. So there are examples of S.R. Chaudhary in Simlipal, example of Billy Arjun Singh in Dudwa, rearing a tiger and tigress and leopard. Even Kailash Sankla, who was the first director of the Project Elephant, reared a tiger in Delhi Zoo. He was director in Delhi Zoo. So very enthusiastic practitioners were trying to know about the animal ethology, that to confine to charismatic species. Very late, when Scheller came and wrote Deers and Tigers, that too he was looking into the interface of the domesticated animals to the wildlife. Some kind of research he started. So when you see up to 80s, research is nowhere to shape anything in policy or statute. It was all coming from the top. Even the policy which we brought, whether it's related to, any kind of policy. Even 1975, when we thought that we have to conserve gharials, even then there were some research which were used. I would say that rearing captured egg in captivity and trying to release hatchlings, yearlings back to the rivers. But that was more of a practical experience trying to convert into action. The research has got impetus, I would say, in late 90s or 20s. When the Wildlife Institute of India came into existence, people started doing research and collecting data for primary data for research and other things. But still, I feel both in forestry and wildlife, we have not reached to a level when the research is getting translated into any kind of policy and action. So the topic which I have been given is about this issue, that whether in the field of wildlife we are using the knowledge acquired after research in any kind of action or policy framework. Now I'll tell you that why should we discuss about this. I'll tell you a very interesting thing. In 2019, 3 people got Nobel Prize of Economics. You might have heard about that. Abhijit Banerjee, Esther Duflo, and Michael Kremer. And the economists, they got the Nobel in the field of development economics was the work which they did, and what we call today is a randomized control trial. You might have heard about it. And people who are doing research know about the randomized control trial, which it is called RCT. RCT used to be a research methodology very common in medicine. So what do? You take different samples, samples of the population, and give different treatment to them. Different kind of treatment, and some may be the control, what you call, and some may be a population which are given the treatment. And then you compare that, what happened to the control and what happened to the sample which was given treatment. This was not being done in the field of economics. So development economists, they started doing it, and they researched for more than a decade. The Esther and Abhijit work is basically one paper which has taken 10 years of research in economics. I don't think that anyone in the wildlife is doing a research which has taken 10 years, or data of 10 years, and then telling that, look, this will happen if you do this, or like this. The second part of it, so the Nobel of 2019 was, as I said, was divided amongst Esther Duflo, Abhijit Banerjee, and Michael Kremer. Half of the money went to Michael Kremer. What I'm saying is not written anywhere. What I'm saying with the experience of myself. What did Michael Kremer did? Presently, he is in University of Chicago, which is a institute of repute in economics. Earlier, he was in Harvard. He was doing a research in Africa, and he wanted that mothers, the house, what should I call, the women working in the household should get time to do something, maybe pursuing their hobby, maybe doing some extra work to get more livelihood or to earn more money. But he saw that they don't get time to do anything, either to pursue their hobbies or to carry out any additional work. And he was not knowing that why it is happening. So he started asking people that, why don't you do something? They said that we have kids with us, and someone is crying, someone wants food, and someone was just, throwing tantrums and something like that. So we have to manage kids, and we can't do anything. Then he tried to understand that why don't these kids go to school? Then he came to know that kids are not going to school because they get sick. They fall sick, very frequently fall sick. And they fall sick, then they remain at home for many days. So he tried to understand the issue that why are they falling sick? He came to know that they are falling sick because most of the time they are having the loose motions, stomachache kind of problem. Tried to understand that why it is happening. He came to know that they are playing in soil, and so they have all, that worms in their stomach. So, and that creates problem. You know that the worms are very common for the kids, in the kids. So he thought that let us deworm them as you do the deworming, and see that what result it gives. So he started deworming kids, and when you give the pills of deworming, it comes out through stool, and you become healthy, and whatever you are eating, you become more strong. Slowly and slowly, the pattern of the sickness started changing. Kids started going to school, and the mothers were getting time to do extra work or something. He became famous for his work for deworming. And whatever money he earned and trying to mobilize and doing deworming to the kids instead of writing papers. And papers was written on his work, that how asking right question or doing some very small, tweaking some very small thing can change lives of the people. So when we gather for this kind of deliberation, everyone is thinking that whether your paper is coming or not, what did you write, your paper came into what impact factor, how many citations you had, how many H factors, H index you have got, how many conferences you have attended, doesn't have any meaning. Yes, they have the meaning in references and other things, and you will be remembered for years. I mean, when for hundreds of years your work will keep you alive, which I say that what is the importance of being a researcher. But the time has come that we have to look at this aspect, whether the research which we are doing is going to change the conservation domain or conservation ecosystem or the conservation requirement in the country, and whether it is going to help people or not, whether they are going to help the species or not. So in many fields, when you see, even in the field of the environment side, if you see the economists which are working in the field of like Michael Greenstone and all, everyone talks of, there is a buzzword which is called evidence to scale, evidence to policy, evidence to action. But in the field of forestry and wildlife, this word is nowhere. And we don't know that whatever we are doing, how it will be implemented in the field. And there is no space left, I would say, in the field of science that practitioners can write a paper. Writing a paper, I mean, even doing research is one set of things, and writing a paper is second set of things. So the time is that we have to look at this cross section, that how research can help in formulating policies or helping practitioners to take more informed decision and actions. There are many examples where the research is playing very crucial role. Recently, in 2022, I would say that we started a program which is called, we say, the DNA profiling of the captive elephant. You know that in Project Elephant, one of the aspects of the Project Elephant is to take care of the health and husbandry of the captive animals. And you might have heard that people say that wild-caught elephants are mixed to captive population, and many a times a tusker becomes a makna and a male elephant becomes female when get transferred. Because it used to be practiced, because one elephant will be exchanged with the others, because ownership can only go to the to air, or when you transfer it to someone else. So the only way is that you have the profile of all the captive elephants, so that when you move from one place to another, you know that this animal is is the same. So you might have not seen it, but the rules which have come for the transfer and transport of the captive elephant from one place to another has a provision that they must have the DNA profiling of the animal done. And there is an app for that, which is called Gaj Suchana app, and we have the data of almost 2,000 captive elephants across the country, and almost the DNA profiling of more than 1,000 captive elephant done, and the morphological features of these animals also done. So, I mean, if you have the data of 2,600 animals, say, in next one more year, then mixing wild-caught elephant into the captive elephant will be difficult. So, the tool which is available can be used in stopping the capture or illegally capturing the elephant and mixing to the captive population which we have in our country. So similarly, there may be many examples which tells that the research can lead to a new policy or help in formulating policies. And it is the best time when we should recently, when the Honorable Prime Minister took the meeting of standing the National Board for Wildlife at Gir, he said 2-3 very important things which is also related to his research. One thing he said, that we should have a set of information which can give you an insight that what are the best research done in certain fields of wildlife. So we have started working on that and tried to collect the best research done in certain fields of wildlife conservation. Similarly, the field work on which we have also done some research done in India, say in last 30 years, which can be replicated other place of the country. Third, he said that we should have our own standard of research done and portray them and to display them to the world that, look, these are the works which we have done and these are the best works which we have done. So this is, when you are here and listening people, you should also think in this direction, that if you are doing some research, if you are reading some research, how it can be used in policy formulation. But still, I mean, because still we do not have any, I mean, not having, I would say, I should not say any example, but very few examples where we have taken help of research in taking policy decisions. Recently, I don't know whether I would like to meet many of my co-researchers who are co-authors, but I have never met. I don't know whether it happens to others also or not. But this is happening with me. So I know whether Dr. Bilal is here or not. Dr. Bilal is not here. So I would request all of those co-authors, if they are listening here or somewhere else, and we are common co-author, kindly meet me, because I have heard your names, but I have never met you. For last 3 years, we are collecting data of human-elephant conflicts. In 3 states, we have collected the data, and in 5 states, the researchers have started collecting data. By now, we have analyzed more than 6,000 deaths of humans and elephants, which are having the time span of 23 years, and with all spatio-temporal features. So we wanted to know that if we have to know about the human-elephant conflict, then we should know that which animal is dying, whether males are dying or females are dying, when they are dying, when they are going out, whether there is a seasonality, whether they are coming in night or not, whether the tuskers are going far or the herd is going far. If the herd is going far, that how much, what was the size of the herd, and all kinds of questions which you can think of. We are trying to analyze them and see that if we can derive some pattern, and then trying to see that what strategy or what policy instrument which we can think of taking action in the field. So I personally feel that there is apart from having the academic research loaded with the statistical treatments, there is also need of research which is for the practitioners, or which can easily be understood by the practitioners. And it is the right time when the research is used for all purposes which we call evidence to scale, or evidence to action, or evidence to policy. This is what I wanted to advocate for and speak for. Sorry for not having a structured presentation. Thank you very much. If the floor is open for the questions. So is there anyone who is my co-author? No one. All are in field. Nothing? Yes, please, sir. Suneet, you don't have any question?
Thank you very much, sir, for the excellent talk and the suggestions. You see, I am a university professor, and I have been involved in many funding agencies with DST for large number of years. See, there are different kinds of projects come. Sometimes somebody has an interest in wildlife. It's not that it may not have a conservation value. Maybe in the long run, probably everything has. But sometimes in the short run, if somebody is interested in studying the structure of robin song structure. Now, you can't immediately say what is its conservation value. There is a lot of science and scientific understanding behind it. See, that is one kind of thing. So, organizations like DST, for example, now it is ANRF, they are mostly interested in funding that kind of research. But then there is also, because all of us are trained to do ecological research, all of us, our students are trained to do conservation research. So, I would request that the ministry and even state departments should come forward more and more for commissioned projects, not rather than only the sponsored projects. If there are commissioned projects where you have a mandate, you tell what your requirement is, and you tell us what to do. And then you would see, then we have one-to-one interaction also. Then you have expectation from us, we know the methodology, what to do, and we would do it together. And only then I think we would be able to produce a lot of such research which would have a direct conservation and management value and application.
True. Totally agree with you. My response is that I'm saying that since beginning we are doing like this. I mean, policymakers are saying that do this. As I said, that we decided in Project Elephant that we'll do like this. But my urge is that we have to start thinking that everything which we are doing, even if this is a research, maybe ecological aspect or animal ethology, or anything about structure or function of an ecosystem, which may not directly lead to policy or action, but from across the table side also. I mean, finally it reads that the policymakers have to say that you do research for this, but it has to come from that side also. If I am doing some research, how this research can be scaled up, how this research can be used in capacity building or in policy instruments or in taking decisions. This interface has to be more stronger and closer. You are absolutely correct. Yes, madam.
Yeah, so I mean, I think that's a great point, and you also made a great point. I'm just curious whether you really, whether you and others think that there's a lack of will because being more on the side of research, a lot of young students, a lot of researchers, they really want their research to have impact. I think there may be a impression that people are obsessed with impact factors and stuff. Maybe they are, but they also really want, they see how nature is being lost and they want to have, to contribute in some way. So I think somehow encouraging that dialogue, I'm not sure, I'm not in that space, I don't know, but it would be really nice to think of how proactively that dialogue can be encouraged with, as this conference is inclusive with multiple institutions in that spirit.
We can have dialogue for at least half day or 1 day like this only. Really, you come with your researches and try to discuss that, what value they are having for. I mean, this is a very good idea, that we should increase. I mean, this is a simple session. And I'm not saying any great thing, I'm just igniting thoughts. But there may be more structured and more robust platform of deliberating that how we are weighing these two things, I mean, research and policy, and how we can bring them together, and what are the good things which policy has done and has to be supported with research, and what are the research in place which can be factored in the policy. Yes, sir.
Pandit sir, thank you for that. Was a very insightful talk, especially for people like me who teach, because there is a lot of research which happens, and the student feels that once his research is done, his job is over, and does not carry it forward. And I think there has to be a vehicle now for getting these students to be able to carry that forward. I don't think it's a lack of will on their part. They're probably very passionate about wildlife and conservation, but that platform that it is needed for this kind of research to get not just into policy, but into regular action at, let's say, a protected area management plan, and that kind of thing. We need some sort of a cohesive platform, sir. I think we should.
It's a very good idea, sir. True, you are very good idea. Both the things which have evolved in discussion is fantastic. Very good.
Can I say something?
Yes. Yeah. The floor is yours.
Since you are ADG Forest, I want to know your opinion about what about conservation action plan for insects? They're disappearing globally. There's an insect apocalypse. We are not talking about anything beyond tiger, elephant, leopard. So why aren't we talking about insects? They're disappearing. Have you any policy in mind, or is there some existing policy?
I take it differently, a bit. I'll give first my experience. What is happening, if you see the multilateral environmental agreements, what are they? One is CITES, I said. There is another CBD. There is another CMS, Convention for Migratory Species. Then there is SDG goals. Then there is something called this global forest goals. Then you might have heard the Cumin Montreal protocols. In many countries, including India, there are different set of people working in different fields. We are more obsessed with protection, habitat management, and working on larger species. If you talk about, say, species conservation, we have a list of 21 right now. Today we have increased 2 more species, gharial and sloth bear for conservation. But we don't have anything to do with hundreds of other species. They are part of the ecosystem. If they are being protected, it's good. If they are not, we don't know about them. But there is another set of people, which is called, say, NBA, National Biological Authority. I mean, they are, and most of such creatures are not confined to forest. Many of them are having a very close association with the other land uses, especially the agriculture. And we had a system of calling many insects as pests. It's the same society which look at these insects as pests, or we call exotics, for instance, or we call endemic, for instance. So, I mean, we treat the same creature, which may be a good biodiversity for you, best for an agriculturist. So similarly, it happens within the conservation practitioners. There are different domains. For me, as a policy maker and as a practitioner, big species with big size, big biomass, may be looking important. What we call flagship species, ketone species, engineers of the ecosystem, rest will be taken care of. Then there is a set of people who are thinking that these are pests or exotics or weeds. So yes, you are correct that many such species, hundreds of such species are not in our targets, because the system which we have evolved is landscape approach. So whatever coming in this landscape is we are trying to protect, and for this conservation and protection, we are focusing on the ketone species, or which is on the top of the pyramid. So if we are taking the species which is on the top of the pyramid, the bottom of the pyramid is taken care of, the food web is taken care of. This has been the approach. And practically it is not possible in country like India or any country which is rich in biodiversity to focus on each and every species. Many species I do not know, and many species we are discovering every year. You will be the last person to ask a question, Suneet.
So my question is about when you talk about research and linking it with the management, which I've been kind of trying to convey strongly to wherever I go. One of the link that we have actually is through the management plans, where there is requirement of the research which is listed there, and the good chances, good scope of getting researchers from nearby universities to get those research work done. What would be your take on that? Because it's listed there, the chapter is there, but not much is happening. So how do you think that could be promoted, and so that we get information good enough to improve our management practices for the species and habitat in those PAs?
Very nice suggestion, very good. See, many of you might have not understood. I'll repeat what he's saying. Mr. Divakar, if I am correct, Divakarji is saying that, I mean, your name is correct? Okay.
Yes, yes.
We are meeting after a long time, sorry. So what he is saying, that we have a system of having management plan of the protected areas. So people write that these are the fields of research which this area requires, but no one is doing that. So can't we get it done with the local people, local researchers, so that we have some data? It must be done, but we don't have. What Bharucha sir was saying, that we don't have any ecosystem kind of thing for research, or thinking that someone has done some research and wants to take this forward, but we don't have any platform for that. Like we were giving, I heard that many of you were given some award to, I mean, some young grant or travel grant or research grant, similar kind of thing, that if research can support your passion or impact which you want to bring in the society, say, to carry forward that action in the field for, say, 5 years or 10 years. As an independent researcher.
See, sir, I would like to give you an example of our own work.
Okay.
I have been doing wildlife research now for 53 years. See, take one example of lion-tailed macaque is an endangered species. We saved Silent Valley because there are 16 groups of lion-tailed monkeys. In my own research in some place in Karnataka, between Sharavati and Agnashni rivers, I found some area where there were 32 groups of lion-tailed monkeys in one contiguous forest, and nowhere else in the Western Ghats. I almost know almost every group in the Western Ghats today. And that particular place had 32 groups of lion-tailed monkeys, 43 groups of bonnet macaques, 151 groups of Hanuman langurs. St. Patrick, all living in that area. I put 2 PhD students, one research associate later on. In 2003, me and my student, we gave up land to Karnataka Forest Department. This is a potential area for primate conservation, specifically for the lion-tailed macaque. It took a long time. We went and interacted with the panchayat people, with local farmers, started a farmers movement there. And my student H.N. Kumara, who is a principal scientist in WII South India Center, in Salim Ali Center, he would ultimately, we took Mr. Singh, B.K. Singh at that time, PCCF there to interact with the people, with the local people. It took us 20 years. But 20 years later, Karnataka government has declared first time between Agnashi and Sharavati rivers 930 square kilometers area as Sharavati Lion-Tailed Macaque Sanctuary. Wow. Then second example, in 1999, nobody knew anything about slender loris. I also occasionally used to see slender loris. Some by chance I came to know about some slender loris near Dindigal. I went with my students. We started doing some surveys and somebody took us in a village. I saw 13 lorises in one night, which was sort of a record. And for our luck, there was Mr. Udyan. He was the DFO in that place. He is now PCC of Tamil Nadu. And I went and told him next day, I had a project going on in Anamalai Tiger Reserve. I was going there with my students, but we just got this information. We went to that village, talked to that one farmer there. He told me from where he got the monkeys. So we went there and told Mr. Udyan, he came with me one night. He got interested. He said, okay, right now we'll write to the Chief Wildlife Warden. I will forward the letter. Together we would do the surveys. So we immediately, I called all my students. I had 7 people in Anamalai Tiger Reserve. Called all of them there. We started along with the forest department. We published our first article in Oryx. The potential and distribution of loris in Dindigal region. Now it again took 24 years of doing this, again and again, doing this survey, road management plan on the basis of our scientific papers. And then 2 years ago, the forest department then asked us, they asked Salim Ali Center, Dr. Kumara, my former student, you give us a plan now, go and demarcate the areas. Which are the areas which needed to be protected? And exactly on that basis, the map which we gave 2 years ago now, Tamil Nadu Forest Department has declared first time 113 square kilometers of Kadavur that in the Karur and Dindigal district as the first slender loris wildlife sanctuary. So it is not that our fundamental research, basic research has not influenced policy. We have been doing it. We have been doing it for 40, 50 years. What I am requesting is more and more things should come from your side also, where you tell your this is what our requirements are. Can you people do this? Then we can do the things together, which will be probably much more valuable and much more applicable.
Thank you. Suneet, my time was over.
So it's not a question basically, it's a remark and suggestion in addition to what you have just said. I would just like to cite an example. There was recently I saw an interview of a famous entrepreneur. I won't name him. You may guess who it was. He said in that interview that he's a very successful entrepreneur and his company is very successful as on date. He said that recently we saw a boom of startups in the country. There were a lot of businesses going on, but many of them they perished and only few succeeded. And those who succeeded, they did so because their main aim was to address a particular gap in the economy, a gap in the needs of the people. So their business was aimed at making the life of people convenient, right? So with that aim, they started their business, they developed it, and that is why they became so successful. And others perished because their main aim was the profit. Though we also like profit and our final motive is that only, but our research, our business initially started identifying the needs of the society or the people. So my suggestion would be similar is the case with the wildlife and other areas also, and we'll be able to link policy and research only when research emanates from a particular problem that people are facing. Suppose we are talking of human-wildlife conflict. Unless we work on the root cause and we route our research through that channel, it will not be possible to link research and policy. So this is just my suggestions.
Thank you. Suneet is my AIG in the ministry. With these words, I end my talk, and thank you very much for giving me opportunity, and I hope that I could just ignite some thoughts and bring these two topics more closer for your food for thought. Thank you very much.
Thank you so much, Ramesh sir, for your talk that ended up quite interactive, which I think powerfully highlights the importance, the need for connect the bridge between research and policy in order to drive sustainable conservation, which I think is the need of the hour as well. With this, I would like to call Dr. Meva Singh, who is an INSA fellow and a professor at Mysore University, to talk about the Journal of Wildlife Sciences for a few minutes. Over to you, sir.
Good evening, friends. I would just take a few minutes, and I'm sure most of you already know that we have a journal recently started called Journal of Wildlife Science. And if some of you do not know, then I want you to know that we have now a journal which is dedicated to wildlife science. This I want to tell you is number of things about this journal. Number one, this is a very unique journal in the world. Why I'm saying in the world? Pick up all of you publish journal, different researches in different journals. As of today, there is no Journal of Wildlife Science anywhere. You have conservation biology, biological conservation. You have ethology, International Journal of Ethology, ethology, ecology and evolution, behavior, animal behavior, behavioral ecology, behavioral sociobiology, behavioral ecology and sociobiology, Journal of Wildlife Management. These are all journals where we all publish. Okay, more natural history oriented like Oryx or generalist journals, but there is no journal which is fully devoted to wildlife science. So this issue we have been discussing for decades together, and finally the Wildlife Institute of India has come forward now for us to publish this, and Ministry, MOEF and CC has generously given enough funds for us, and I'm the editor-in-chief of this journal. We have started this journal, and the idea of this journal is to publish basic scientific research on wildlife. All different topics, it is with the basic science, it is with conservation, it is with management, everything which is related with wildlife. So the scope is very wide, but the most important thing I want to tell you now is you are aware of different ways of publishing nowadays. There is one way of publishing like the Springer or Wiley or Blackwell or any of these people. They generally have a model that the authors do not contribute, but the readers have to contribute. Usually $39.95 is you can one time download an article, which many of us don't do because our universities or institutions have subscriptions. So that is one model of publication where the readers pay. Now you have other mode of publication like all PLOS publications, MDPI or whatever, all these publishers is where the authors have to pay the money. It is open access, called open access journals. No, the people, the authors pay a lot of money, and it's not small money. Imagine people like us, what would happen to our future students? Every journal now, PLOS used to be 1,000, 1,500, 2,000, 2,500, global ecology and conservation is 2,650, scientific reports is 2,500. I mean, this amount of money, from where our students would pay this kind of money? So that is another way of, mode of publication. Now there is something third mode called diamond open. Let me tell you that we have now not a golden opportunity, but a diamond opportunity that we have a journal here which is totally open for readers as well as authors. We do not charge any money from the authors like most of the journals do. We do not charge any money from the readers who have to pay the money to download articles. So this is a journal which please go to this site, JWLS, Journal of Wildlife Science, and we started it last year. We have brought last year one, full issues of the last year. We are into second year. We have been already getting good number of articles. As we just had an editorial board meeting, we are very choosy about the articles, although when Bilal, Vishnu and me, we wrote the first editorial, we wrote about this that look, our emphasis is not like rejection. Many journals take pride in saying, we reject 70% articles also, although we are also doing, but then we have a different mode. We openly have told that, look, our emphasis is to encourage people for publication. We have written it openly that it's a blind, peer review process, but if the author and the reviewer agree to it, we would like mentorship by the reviewers. We would like the reviewer and the authors even not telling us, they can exchange information with each other. So we are trying to develop that kind of models where we try to encourage people and help people, mentor people for publication, and all free of cost. And we have all different kinds of topics covered. There are articles coming on flora, there are articles coming on fauna, of course, wildlife, and different type. I am a hardcore wildlife biologist, a field biologist, but because I have done a lot of work on captive breeding in Germany, in the US, with my colleagues, I am now writing series of articles about what has happened for 100 years in the zoos, what has been the problem of conservation breeding. Another article on zoos in India, 50 years analysis, what has been going wrong. So we have all these kinds of topics covered. So I encourage all of you people to please go to this site and submit it. And we can also have special issues if somebody has an idea that you can contact different people. You know different people, you have a theme. If you can get 6 or 7 articles minimum, we can also bring out a special issue. We would also offer editorship to somebody. Somebody says that, look, a person may not be on our editorial board, but if a person says that, look, I can bring you 6, 7 very good articles on a particular theme, we would offer, okay, you be the editor. The journal will be under your name as an editor. We will offer it. So this is a very flexible kind of situation, an enormous amount of opportunity. So please make use of this opportunity and submit for this journal. Keep this journal in mind, and please also spread the word about this journal to other people. Thank you very much.
Thank you, Meva sir. It is time for our second spotlight speaker, second spotlight talk for today. So it is my pleasure to introduce our spotlight speaker for today, Dr. Jitesh Jhawar, Assistant Professor at Ahmedabad University. Dr. Jitesh earned his PhD from IISc Bengaluru, where his work on schooling dynamics in fish revealed simple rules behind complex group behavior. Dr. Jitesh's work focuses on collective behavior and self-organization in animal groups across birds, ants, and fish using machine learning. After his PhD, he continued his research at the Max Planck Institute of Animal Behavior and the University of Constance, supported by prestigious Alexander von Humboldt Fellowship. He is now setting up a lab to explore collective dynamics in natural and semi-natural habitats. Please welcome Dr. Jitesh.
So, a very good evening to all of you, and heartfelt thanks to the organizers for giving me this opportunity, a very attractive title to this session. So my talk is going to be different than most of the talks that we have listened, in the conference so far. So I thought I would spend a few more minutes in, to the motivation and for my work. So I thought what would work better than putting this paragraph, quote unquote, by the famous physicist Richard Feynman. So please allow me to read this. So everything is made of atoms. That is the key hypothesis. The most important hypothesis in all of biology, for example, is that everything that animals do, atoms do. In other words, there is nothing that living things do that cannot be understood from the point of view that they are made of atoms acting according to the laws of physics. This was not known from the beginning. It took some experimenting and theorizing to suggest this hypothesis, but now it is accepted and it is the most useful theory for producing new ideas in the field of biology. The study of patterns very nicely aligns with this word of thought, because patterns are everywhere, right from molecules to galaxies and our animals. They show remarkable patterns, fascinating patterns. Across taxa, collective dynamics is omnipresent. As you can see, right, starting from a fish school to insects showing very interesting behaviors like ant bridges, and mammals showing really nice, herd formations. So I would like to show you some more videos from my own work from the past, and just to show you, sort of get going into the study of patterns. Here is a school of fish which is swimming nicely in an experimental tank. This is a school of, this is a group of, a swarm of beetles that are showing us very interesting pattern called milling. You can see, and I have sort of spent sometimes at least half an hour just looking at them. They're very, very miraculous to look. And of course, we have swarms of honeybees, 2 flocks of starlings. This is a video captured in my university. They come every winter and just mesmerizes us. And there are interesting behaviors by insects, including ants. Here, there is an example of something called cooperative transport. I'm sure all of us have seen them, coordinating with each other to carry larger or heavier loads. And so this is my motivation to, do what I do. And the question is now that how can we study collective behaviors and group dynamics? So one way through which people have been looking at group dynamics or collective behaviors is through this question of the proximate or the how. That is, how do individuals interact with each other such that we see these interesting, fascinating patterns that we are observing. And the other is more sort of ultimate question of why do animals come together? Why do they form groups? Why do they live in groups? There are many benefits that have been hypothesized, tested, and found, such as some alarm calls or dilution against predators, some mechanical benefits through flights, and so on. So my primary focus has been to study the interactions part, which is how do individuals interact with each other such that we see these interesting patterns. And in this talk, I'll be talking about 3 different systems. One is Heteroplus suratensis, a cichlid fish from South India, and then we have whirligig beetles, which you saw the video, and then, of course, honeybees. Within these 3 systems, I'll describe studies from totally controlled, totally undisturbed experimental environment to semi-natural, as well as now we are sort of moving out into more into not really into the wild, but into the campus, whatever wildlife we have. And with, of course, we'll see some experimental perturbations to study the response of these collectives. And we use a blend of experimental as well as theoretical approaches. and that's what I'll talk about. So to begin with, people have been really interested in collective dynamics from long time. This is a famous model from 1995, which described or which was successful in producing interesting patterns or recapturing or patterns that we see in the wild, such as schooling or milling patterns. And the very simple, the model is based on very simple assumption that individuals follow the behavior of their neighbors within a certain vicinity. And I'll skip what all these models have found because they've created very interesting predictions for the experimentalists to test, but I'm not going into that. Instead, I want to talk about something that I was interested in when I really started this or getting interested in this all field. So I wanted to use a mathematical framework to study collective behavior. And to get into that mathematical framework, I would like to touch or go back to the more sort of known equation to us, which is the famous logistic growth model that I'm sure we all have seen before, or we all can see not just in equation but in the real world, that populations grow and then populations saturate, and after some time populations also decline when there is limited resource. So a very simple model can capture this, which is written here. It's basically the rate of the growth, n dn by dt, is dependent on the current population size, and then there's a factor 1 minus n, which denotes that the carrying capacity here is, we have just put equal to 1. So if the population starts anywhere, it always will go and saturate at this point k equals to 1. This model can be, or this result can be understood from another perspective, which is the per capita growth rate here. So instead of writing it in this form, I've just put the n here, and you can see this is model how the per capita growth rate varies is basically a linear function, right? So what is required at this point is just to understand that stability can be understood in terms of either the population size or also the per capita growth rate. And we will, I would like to focus on this point here. So whenever, before the, when the population is before 1, which is the carrying capacity in this model, the per capita growth rate is positive, okay? And whenever the whenever the population is above 1, the per capita growth rate is negative. So what this basically means is that whenever the per capita growth rate is positive, the population as a whole is going to grow and grow and grow. And if it reaches 1, it will stay there. If it shoots above, it will come back. So this point is called a stable equilibrium or stable fixed point. So this is enough theory for linking or understanding how can we apply this known framework to study or model collective systems. So basically, let's consider a very simple framework here. So assume that, let's just assume a very simple framework where animals or individuals can move only in one dimension, either left or right. Okay. And then there are certain assumptions here. For example, the group is always cohesive, which means that everybody can interact with everybody. And these are some very simple rules that we will allow in the model to happen. So basically, these rules tell how do individuals interact with each other. So one rule is very simple, that from a given state, individuals can be in any given state at a particular point of time, either left or right. So from that state, there is a spontaneous transition to the other state. So from left to right or right to left with an equal probability. Then there is a very simple interaction between the individuals, which is that if there are two individuals in opposite direction, after the interaction, they start to move in the same direction. and this is a symmetric interaction. So, if the individuals are in the opposite direction, there's equal probability to transition into either the left or the right direction. So given that these two rules, we can write a similar equation as we saw earlier of the collective dynamics itself. And that I'll be telling you about. How to arrive at that equation is in a very concise manner. So here, we assume at any point of time, there are x1 individuals in the left direction, and then x2 individuals in the right direction. So if we take the difference of that, basically denoting it by m, and then m is equals to x1 minus x2. So at any point, if there are equal number of individuals in left and right, m will be 0, which means that there is complete disorder in the system. On the other hand, if all of the individuals are either in either one of the state, either left or right, m would be either 1 or -1. Okay, so this m is also known as the order parameter in the system because it describes how much order is there in the system. And by order, we can also call it collective order because we are talking about a global property or the property of the collectives. So the interaction that we saw can be written down in the form of chemical reactions as well. So we use a chemical reaction framework to model this. So the first two interactions are the spontaneous transitions with a rate S, which is the coefficient of this rate, and it depends on how many number of individuals or proportion are in the x1 state. Similarly, for x2. And the copying reaction depends on the proportion of both the individuals in both the states. So if either of, let's say, no individuals are there in x2 state, you can imagine that there won't be any copying from the x1 state to the x2 state. Okay. So this equation basically allows us to plug in whatever we have here, that is the rates and the coefficients by which each reaction changes the proportion of individuals from one state to another. And basically, we arrive at this equation that was our objective to model the collective behavior. And the equation looks something like this. M is the collective order or the order parameter, dm by dt. The rate of change of the order parameter is equals to minus Sm plus root over 1 over root n and under root there are some coefficients. And there's a eta t term here, which basically is the noise term here. And I would be talking about this noise term for the rest for the next 2-3 minutes. So, but before that, to analyze this equation, let's just forget the noise term for now. And we are back at the simple equation that we saw earlier, which was a linear equation in a variable, right? So here, basically the rate of the system depends changes negatively with the system state itself. And just like we had a stable fixed point at some k value, here we have a stable fixed point for m is equals to 0. So the model that we have derived from the simple equations, simple interactions, is actually telling us that the system will always reach disorder deterministically. But then when we look at or when we simulate or find the solution of this model, it actually gives order. It actually produces order. So what is happening is that because we actually neglected the noise part, so we have to look at it again. So the noise part is actually here very interesting. So the noise itself here depends on the state of the system. So unlike traditional models where we add noise in an ad hoc manner, here we are finding, or from the model, we are finding that the noise is actually multiplicative because of this, multiplicative term in front of the eta. And to understand this, basically what is happening whenever the system is reaching the disordered state, the fluctuations in the dynamics are very high, which continuously kicks the system out of the stable potential of disorder, and then it's continuously transitioning it to the states of high order or finite order. So this is also known to be something in the physics literature, but not that common. But from the collective behavior perspective, what we find here that stochasticity or noise can induce or create new states of order. And we also find how to link individual level behaviors to collective dynamics here through the use of equations. So here we can see that the spontaneous reaction that we had modeled actually turns up in both the deterministic as well as the stochastic term. But interestingly, the pairwise copying interaction that we had put actually doesn't end up in the deterministic part at all. Instead, it is actually in the noise part. So it turns out that it's very important to actually look at the noise in the system. And there is, of course, the 1 by root n, which is, basically tells us that as the system size increases, the effect of noise decreases. This is also kind of similar to the demographic noise or genetic drift as well. So when basically that tells us that as we increase the system size, the effect of noise should go down, and we should reach, or we should basically, find the system to be in the deterministic or show deterministically stable states. And this is what we, get. So now we have looked at briefly at what collective or what models can tell us. It's time to look at the real systems and actually see, if such interesting properties also exist in the real world. So we'll look at the two systems that we have already talked about. When we started doing the experiments, we actually, the question was that how to now, whatever we are seeing in the model, how to compare it with the data. And the traditional approach is that, we sort of find the solution of the model, we find, some interesting property of the data, and then we compare. However, here instead, we actually derive the kind of equation that you saw for the group dynamics. We actually derive that equation right from the data itself. And once we have this equation, now we can go back to an individual-based model framework and actually compare if we are getting the same equations from both the frameworks. So, and then hopefully infer the underlying mechanisms. So there's a package now called PyDaddy, very interesting name. It stands for, I think, Python package for data-derived dynamics. And it is now open source. So basically, if you have a long time series of data, you can just put it in, and then it will, throw back an equation at you that you can analyze. Of course, there are a lot of nuances that one needs to take care. But essentially, that's what it does. It is available to download from any Python environment and so on. So basically, that's the idea. As I said, now we want to look at how do the, are these properties, interesting properties also there in the real life or real systems. So we have a system here, which is fish, Atroplus uratensis. So we studied it in the lab in undisturbed, totally undisturbed environment in very controlled manner. And we only varied the group size from 15, 30, and to 60. We recorded for about 60 minutes, and for each group size, we had multiple trials. Compared to the size of the arena, the size of the fish is pretty small. And we keep the depth of the arena or the water to be low to create some two-dimensional environment, so that there are no confounding effects of the depth as well. We use some computer vision techniques, and at that time, during my PhD, we used some in-house codes. But now, with the advancement of technology, these things can be done much, much more easily. So So the idea is that once we have these experiments and we have our framework, how to quantify the behavior. So we'll basically, the order parameter that I described before for the model, here also we used an order parameter to quantify the behavior or the collective dynamics. Here it's called a vector polarization because the system is 2-dimensional, and then basically, for describing any system in 2 dimension, that can be done using 2 vectors. Now, but we can always get back the scalar order parameter, something similar to what we saw earlier, by using the magnitude. So here, whenever, basically again, when mod of m is 0, it denotes disorder. Whenever mod of m is 1, it denotes that the school is highly aligned and moving nicely in the arena. So what do we find? As the group size increases, the group size 15 shows high order, as we can see in the histogram in the bottom left. There are high peaks at near m equals to 1, and a very low densities near m equal to 0 and so on. Also, we can create a 2-dimensional histogram of mx and my, and these high boundaries or densities at the boundaries tells us that there is the fish basically, the school is basically spending a lot of time in a very highly aligned state. Okay. Now, as we increase the group size, we find that the order in the system reduces and reduces, as evident in by the histogram, as well as also when we look at the time series. Okay, so the result is pretty simple and neat, that as we increase the group size, the order in the system is reducing. Okay, so just we can recall to the model results that if you know that when we increase the group size, there was actually disorder at the higher group size, and are the patterns of the underlying dynamics also holding in the data? It is in fact the it is in fact happening. So as you can see through these projections, we derived the model from the data, and it turns out to be exactly similar, the functional form to be exactly similar to the very simple model of pairwise interactions between individuals. And this basically the model captures the salient features of the data, that is, as we increase the group size, the order reduces, as well as it captures the underlying dynamical patterns. This is very important because we have also shown in another work where very similar models can produce, very different models can produce very similar dynamics actually. So since the model was in 1 dimension, we actually show this in a 2-dimensional model also through a continuous angle model, because earlier I just showed you that there is very less degree of freedom, right? So this model again captures what we find in the data. So to summarize this part, we find a very interesting evidence of the noise-induced cooling, and supporting the idea that noise needs to be looked or dealt more carefully than traditional methods. We find very simple pairwise interactions in the model can explain the noise-induced effect that we observe. And the intrinsic noise turns out to be actually the informer in our work for discovering underlying behaviors. So moving on to the next system, which is also very interesting. It shows very beautiful patterns of milling when we put them together in different group sizes. And here, the order parameter is a slightly, is slightly different. Instead of a linear polarization, we have something called angular polarization, standing for milling or circular motion. So whenever m is equals to -1, it represents clockwise motion. Whenever it is +1, it represents counterclockwise motion, and whenever it is 0, it represents the beetles are of course moving, but no net angular order. So very quickly moving through this, as we increase the group size, actually here this time we find that the beetles become more and more order. The swarm becomes more and more circularly polarized as we increase the group size, as evident from the histogram densities here, as well as the time series. You can see that there are spontaneous shifts from state of counterclockwise milling to clockwise milling and so on. However, the time series here shows that the system is mostly disordered. So the data-derived model, again, using the same framework, so these are the functions that we derived, and then the fits over them. So using this equation, we actually analytically derive the densities from the model, and you can see how nicely they capture the behavior or the quantity, the behaviors that we are observing. So, so with that, we also test a spatial model here, because as the idea is to find or to discover the individual level behaviors. So we also test a classic individual-based spatial model, and we explore this, and we find that the model actually describes what we are finding in the data, that is from order to disorder. So what is the difference between the previous model and this model? Because the earlier model was actually capturing what we found in fish, and this model is capturing what we are finding in beetles. The difference is the interactions that are present, or that are being plugged for each individuals to interact. So here we have to actually plug some complex interactions. So here, because of, as you can see, the drift function, instead of just being linear, the drift function is actually cubic. So it turns out that we have to incorporate some complex interactions to capture or cover this dynamics that we are observing. The spatial model we tested for a bunch of group sizes, and qualitatively it does a good job in capturing the features, right, all the features from the drift to stochastic, as well as how the order changes with group size. So, so with that, I think this part I would like to just conclude that we present an example of how to, how we have sort of integrated data with models, and how the models can lead us to inference of behaviors. And moving on to the next system, which is honeybees, and the system, the experiments are different from the previous two works in the sense that here the honeybees are kind of interacting with the environment throughout the experiment, because there's an observation hive, although it is semi-natural, so observation hive is sort of in the room, but they are continuously interacting. And we also study the effect or how a collective system can sense the environment as well as some perturbations to the environment. So inside a honeybee nest, we know that there is a queen, and then there is a reproductive division of labor, and there is a non-reproductive division of labor amongst the workers. I'm really fascinated by how the non-reproductive division of labor works, and how all the workers coordinate their behaviors. So here I got a chance to work on this data set of these very, very intensive experiments conducted over a span of 2 years. For every year that the experiments were run for around 3 months, and continuously for day and night, and recorded, and then fed into a computer, and the computer would automatically analyze the positions of each and every individual. So it's really a very, very powerful data set. And how do we get this position of each and every individual here? By putting these QR code tags on each and every individual. Actually, not every. So there are around 10,000 bees in the observation hive. So the configuration allows us to put 4,000 tags at maximum. And then there is an automated system which can recognize the entire trajectory of the bee throughout its lifetime, from the moment it is introduced into the observation hive from a very young stage, young adult, to until it vanishes either in the wild or inside when it dies. So very quickly, moving on to, there were certain experiments. So the first year of the observation was pretty much for a baseline, but in the next year, certain experiments were done. I was part of one of the experimental data that I analyzed, which was putting the hive through a heat stress. So basically, the bees like to be in an ambient environment of around 30 to 35 degrees Celsius, but the temperature was raised almost till 40 degrees Celsius of the room. And then there were also temperature sensors in the hive, which shows that the hive temperature actually increased as a result of the temperature increase in the room. And these frames show the map of the nest itself. The different colors denote in what part of the nest what substrate is present, because the bees store honey, there is brood somewhere, there is pollen, and so on. So this mapping was done manually every 5 days. And as a result of the increase in temperature, there is response from the colony. The brood, the number of individuals that were taking care of the brood, they actually reduced. So if you just look at here, the honey is actually on the top of the nest. So they move towards the top where the honey is, and pretty much the fraction of bees that are also outside, that fraction also increases. So they also move outside. And then in terms of the movement matrix, the speed of the bees, they are sort of just moving very fast. They're dispersing too much. And so this is the colony level response that we observe. That is primarily, a bunch of them are moving out, a bunch of them are moving away from the brood. So the question is, can we now look at the individual level, what each and every individual is doing inside the hive. And so how we do this is, because we have the behaviors of each and every individual, a multitude of behaviors, as as I showed in the last slide, that is how much time a particular individual is on a particular substrate, or it's spending time outside, what is its speed, and so on. So we can use PCA, principal component analysis, to classify these behaviors, and then project back onto the whole population to understand if we can classify behaviors into principal or dominant behaviors. So this is what we do. Basically, we classify the behaviors before the perturbation, and then we classify after the perturbation. Before, we find 5 primary behavioral clusters, and then after the perturbation or during the heat stress, we find 4 behavioral clusters. Principally, you can see that the M3 cluster, which is primarily on the brood, that is absent in the primary clusters during the heat stress. So with this, because we know what each and every individual was doing before and after, we can actually map how many of, for example, M1, the bees which are in the M1 cluster, how many of them actually transition to H1, to H2, H3, and so on. And that's what we do. So before, we have these multitude of behaviors from the clusters, and during the heat stress, we have these. And this heat map very nicely describes the transition. So this is nothing but a transition matrix of behaviors. And just to summarize, individuals respond to heat stress to mainly avoid avoid any causal effect because of the heat stress, either through their own body heat or something. Therefore, they move away from the brood. That's the dominant response. Individuals that move away from the brood, they prefer to stay deeper inside. And individuals that actually vacate the nest, they are the ones which were already transitioning between, or making trips, foraging trips, and so on. What we do not know is, for example, if those individuals were doing foraging trips, do they transition to do water trips now, because they have to collect water to bring inside to cool the nest. So I think this sort of behavior, a coarse-grained look at using technology can be combined with traditional methods of observations to find out more about what is really going on. How do these large colonies actually handle stressful situations? So I'm currently at Ahmedabad University, and this is a small group of the lab. And now the focus is organically moving towards, I think, the why part as well. Why do individuals form groups and so on. So we are doing some new preliminary work where I'm just showing you some fresh results here. So this is a work where, in the campus, we get lots of bird flocks, and we started looking at them primarily from a mixed species aggregation or mixed species grouping perspective. And as we know that, individuals gather to collect various benefits such as many eyes effect and so on. So we wanted to just test whether these hypotheses also hold in the urban context. So that's what we are looking at, and we chose 3 different sites within the campus, and with varying disturbance, degree of disturbance, and we find some interesting results. The site which is least disturbed has the most diversity. The site which is highly disturbed has least diversity, but it also has large number of, the group size is large. So lot of aggregations, but less diverse. And in terms of what are they doing, the site, across all sites, we find that the foraging more or less increase, except site 3. The foraging pattern, or birds are spending more and more time for foraging as the group size is increasing, and also the vigilance may be reducing, at least for site 1, so it seems like that there within a small sort of area itself, one is that the thing that comes out is, one is that we can have variations in the behaviors, and some of the primary theories may hold or may work, but depending on the context and the level of human disturbance and so on. Another interesting project that Divin started in my lab, when the starlings came, he got very interested in them, and so basically this is something that he came up, that why don't we look at their shapes and start to quantify their shapes as they are forming or they are flocking. So the idea is to basically, the main idea is to understand why are they still, or why are they flocking? Because primarily, it has not been studied. We all see them, but there are some work which have been done on, understanding their interactions. For example, there's a model which says that they may be using some kind of topological rules and so on. But there are some hypotheses that they may be just roosting for the warmth and so on. So I'm not saying that we found the answer, but the results I will show you are interesting. So he classified basically the collective of flocking into different shapes. I don't remember the full forms of all of these, but from something called small road to ovoidal and so on, W-shaped, globular, and singleton. And so one is that he checked as a function of the time before sunset, how do the frequency of each of these shapes is changing. And more or less, there is no distinction. So throughout the whole event, all sort of shapes are fairly common or similarly commonly observed. However, he also analyzed, in the presence of kite and in the absence of kite, basically separated the data. And then it turns out that in the presence of kite, one particular shape becomes very dominant, which is small road. So I don't think I have it here in the photos, but I'm just guessing that it's very linear. It's very, I'm tempted to hypothesize something, but yeah, it's just interesting that we see these patterns, and these are very promising results, I think. So this is another project that has just started, where we are interested in aggregations as well, particularly interspecific aggregations and particularly competition between the birds. And for that, we have set up a feeder in the university, and we monitored the feeder through a CCTV camera and provided some food for, so this happened during the winter from January to June. And we had like continuous data of the the birds visiting the feeder, and so here with this, our main objective is to understand the interactions between different species, and in particular in the face of competition. So, and as well as some perturbations like pressure and so on. So this is really a work in progress, and there's not much to say here. But we are using, as many of you have also shown, very nice, application of technology. So we are also doing that, using machine learning to, first of all, classify different species that are visiting. And the idea is, can we also quantify behaviors eventually? And I hope we are moving there. So once that's sort of done, I think these could be powerful methods to actually study behaviors also in other places or near a, or inside a protected area, and not just to monitor, but also to understand the behaviors. So a bunch of people to thank, and with that, I would like to thank all of you for listening, and would like to leave you with, again, some cool movies. Thank you.
Thank you, Dr. Jitesh, for this thorough overview of your work. Unfortunately, we are at time constraint, so interested folks can contact him personally. He'll be around the campus. Thank you so much, sir. Adya, over to you.
Thank you, Dr. Jitesh and Shrushri. Before I introduce our next speaker, I'd like to take a moment to talk about eye contact bridge. Wildlife conservation has evolved significantly. We have moved from relying on simple tools like binoculars to now incorporating a wide range of advanced technologies such as camera traps, acoustic sensors, satellite telemetry, drones, genomic tools, among others. However, over time, we've come to recognize a persistent gap between those who develop these technologies and the conservationists who use them on field. Icon Tech Bridge aims to bridge that divide by fostering meaningful collaboration between technology creators and the users. As part of this initiative, I would like to introduce Dr. Ravi Kumar Chillukoti, who is the tech support head at MGI. He will be presenting on MGI SEC, All Genomic Tech for Conservation Genomics, an exciting leap forward in how we can apply cutting-edge genomics to real-world conservation challenges. Over to you, Doctor.
Thank you. First, before starting my presentation, I would like to thank to organizers for giving this opportunity to MGI, and especially for Dr. Bilal and also Dr. Vishnupriya. So by the way, I am Ravi representing MGI in this conference. I'm taking care of technical support and also product management from MGI. So in this conference, I would like to introduce MGI's genomic tools that can be applied for the conservation genomic studies. So this is the content of our presentation. So already since last one day, we are discussing on biological diversities and also biological diversity conservation studies, and also then where we can apply these conservation genomic tools, especially for identification of the species diversity and also the genetic diversity together with the natural habitat. So already most of some of the, since last one day, most of the presentators already they have mentioned some of the genetic tools, genetic markers that can be applied for conservation genomic studies. For example, here you can see already the traditional genetic markers, SNPs and also biallelic markers that can be applied. But compared to the single allele and also biallele markers, the multi-allele markers that will be highly helpful for identification of the species diversity and also genetic diversity. So apart from this single allele and also biallelic markers, the multi-allelic markers, especially the SSR markers, for example, short tandem repeats that can be highly accurate compared to single allele and also biallelic. So there are multiple techniques that can be applied for this identification of the SSR markers, SS. And apart from that, the advanced tools now we can apply, apart from the traditional markers like multi-allelic markers also, we can apply the whole genome sequencing studies also. So most of you are already using the whole genome sequencing, but it will be generally very expensive compared to the multi-allelic markers, for example. But now the whole genome sequencing also, it's very low expensive now. So for example, from MJ, you can complete the whole genome sequencing of maybe elephant and also any other animals within 8,000 rupees per sample, for example. So here you can see some of the genomic tools that can be applied for the conservation genomic studies. For example, most of the presentators, they didn't mention about environmental, the species that can be, especially the microorganisms that can be identified using the DNA that can be extracted from the environmental samples, for example, water and also soil. So the DNA barcoding tools that can be applied for identification of any kind of microorganisms from the environmental samples, for example, water and sample. And also you can identify what are the microbes that are associated with the animals, okay? So by using this DNA barcoding tools. So for example, here you can see the 16S RNA that can be applied for the identification of the bacteria, and also ITS that can be applied for the fungi, and 18S that can be applied for the nematodes protists. So instead of using the simple and also individual tools, you can use the whole genome metagenomics that can be identify all the microbes that not only the bacteria, fungi and also and you can identify the virus also that are associated with the animals by using the whole genome metagenomic studies. And apart from that, as I mentioned to you, so the multi-allelic marks, especially the short tandem repeats, which are short and also highly repetitive, and these are highly accurate for identification of the species diversity and also genetic diversity compared to the single allele and also biallelic. So they can be applied for the quantification of the genetic diversity especially, and also they can identify what are the species that are having the inbreed, especially the low fitness. So this way we can use this STR alleles for identification of this inbreed species also. So as I mentioned earlier, whole genome transcriptome that also can be applied for the identification of the virus that are not only the DNA viruses, there will be some RNA viruses that are associated with the animals. So by using the whole genome transcriptome tools that can be identify the RNA viruses also. And if you need to apply the whole genome sequencing, for example, for the large scale of the samples, that will be very expensive. So that's why instead of the whole genome sequencing, you can apply the low-pass whole genome sequencing instead of the high depth of the sequencing, for example, 30x or maybe 60x or 100x. You can apply 5x or 10x. So this way, you can sequence large scale sample volumes using the low-pass whole genome sequencing for the species diversification together with the genetic diversity. So here you can see NGS tools that are being used as a major tool for the conservation genomics. So here in this slide, you can see various NGS technologies that are emerging from 2005 to till date. So MJ has started manufacturing of the NGS platforms from 2018 onwards. Before MJ has started the manufacturing of the NGS platforms, the cost of the sequencing was very expensive. So when MGA has entered into this whole genome sequencing using the technology called DNB-seq, DNB-seq stands for DNA and nanoball sequencing. So now the cost of the sequencing is really driving down. So now we are offering $1 for 1 GB data output using an instrument called T20. So here, by using this one, you can complete the whole genome sequencing of any animal within 8,000 rupees per sample. So coming to the core technologies that are developed by MJ that can be applied for the conservation genomics. So there will be two kinds of modalities for the sequencing. So the short read and also long read. Short read by name itself, it cannot sequence the long DNA molecules. So that's why once the DNA is extracted, the DNA should be fragmented. Then the fragment DNA will be ligated together with the adapter and also barcode. Because if you need to sequence multiple samples, we need to add a tag for every sample. So the tag is called barcode. So the barcode and also the adapter will be ligated to the fragmented DNA. Then the sequencing will be completed using the short read sequencers. So then the reads that are coming from every sample, that will be mapped to the reference genome. Okay? So that's why generally the short read sequencer, they require the reference genome. By any chance, if the animals or any microbe that is not having reference genome, then you need to apply the de novo assembly. So instead of the same reads that are coming from the short read, they can be overlapped together for the construct of the novel genome for those animals which are not having the reference genome. Okay? So whereas the short read sequencers, especially for the de novo assembly, if the animals are having highly repetitive sequences, then it may not complete the construction of the whole genome. So that's why the long read also will be highly helpful for the construction of novel genome for any animal which are not having any reference genome. So here, it doesn't require any fragmentation of the DNA. Once the extraction of the DNA is completed, then the sequencing, we just add the adapter and also then the sequencing can be completed. Then the overlapping reads that can be constructed into the novel genome. Okay? So here, the long read sequencer that can construct any animal which are having highly repetitive sequences also. But sometimes there will be some gaps will be there in the long read sequencer. So that's why some of the short read DNA data also should be applied for the long read sequencing for filling up the gaps. So here you can see the differences and also disadvantages of both short read and also long read. For example, here you can see the short read, it will be having the high sequencing accuracy, whereas the major limitation of the short read is it cannot complete the whole genome sequencing, or maybe it cannot sequence any fragment which is having more than 500 or 600 base pairs. So for example, if the STRs, short tandem repeats, which are having a length of more than 600 base pairs, it cannot complete the sequence. So that's why better to go for the long read sequencers for the STRs which are having major length more than 600 base pairs. Okay? Whereas the major limitation of the long read is the sequencing quality is a little bit lower compared to the short read sequencers. But whereas nowadays there are some companies, for example, PacBio, which are providing quality of the sequence up to 99%. So once the sequencing is completed, sometimes as mentioned by some of the presenters, you need to apply the AI tools and also machine learning tools sometimes for the construction. So here, coming to the core technologies that are developed by MJ for the short read and also long read, for example, here, the DNB-seq technology that stands for the DNA nanoball sequence. This is the technology developed by MJ in 2012. So then we started the manufacturing of the short read sequences from 2018 onwards. So last year, we released the long-read sequencing technology that is named the CycloneSec. Actually, this is released into the market just 1 year back. So the CycloneSec is nothing but the nanopore sequencing technology. So what it will do is, once the DNA is extracted, we just ligate the adapter. Then we use something called, some protein called motor protein. What it will do is, it will bind to the adapter region of the DNA. Then what it will do is, it will unwind the double-stranded DNA into the single stranded. Then it will transfer the single-stranded DNA one base by one base through the nanopore protein, nanopore hole. So then the bases will be identified. This way the sequencing will be identified. So as I mentioned earlier, the quality of the sequencing in the long-read sequencing through the nanopore sequencing technology will be little bit lower compared to the short-read sequencing. So that's why nowadays most of the researchers, they are applying both the combination of the short and also long-read sequencing, especially for the animals which are not having in reference genome, and especially those animals which are having highly repetitive sequences, better to apply both the combination of the long and also short-read sequencing. So for example, here you can see the product line that are designed by MJ for the short-read sequencing. So we have designed multiple platforms depending on what are the applications you need to apply, and also what is the volume of the samples you need to sequence in one run. Almost done. So for example, here, especially for the conservation genomic studies, G99 and also G400, these two are the ideal platforms. Already in WII, the G99 is being used for the conservation genomic studies, especially. So the long-read sequencers, there are two platforms. So the WT02 and also WY01, especially for the whole genome sequencing, the WY is the ideal platform because the data output is 400 GB data output. So here you can see the applications that can be applied for different applications. For example, the G99, which is the ideal platform for the conservation genomics, so which can use the sequencing of any library that is prepared from the multiplexing of the STR alleles. So coming to the sequencing quality, as I mentioned to you, we are providing the guarantee of minimum 85% of the bases will be having the Q40 score or more. Q40 means the quality accuracy will be 99.99%. Please note, 99.99%, which is very, very high quality. So whereas if you see the other company platforms, they are guaranteeing minimum only Q30. So Q30 means the quality score will be 99.9% only. So so if you need to complete a huge number of the samples, check what is the runtime of the sequencing runtime. So for example, if you need to complete the huge number of the samples within the limited time, you can use these platforms. For example, G19 and T1 and T7, these can complete the sequencing run within 24 hours. Especially, for example, here you can see WII, they have recently completed around 2,000 elephant samples within 1 month time on G99. So by using the data generated by the DNB-seq platforms, since last 8 years, more than 11,000 publications have been published in high-impact journals. So apart from the providing of the sequencers, once the sequencing is completed, you need to analyze the data for the huge number of the samples, right? So that's why we are providing the bioinformatics modules also, which are having a hardware called FPGA, Field Program Gate Array, which can accelerate the data analysis of the whole genome. For example, by using the traditional servers, whole genome sequencing analysis will take more than, I think, 2 days. But whereas by using this, our mega board, which is having a FPGA Field Program Gate Array, the sequencing of the analysis of the whole genome can be completed within one hour. And apart from that, if you need to prepare libraries for a huge number of the samples, manual library preparation is very difficult. So that's why we are providing the automation systems also, which can automate all the steps starting from the extraction of the DNA, followed by the library for thousands of the samples. For example, here you can see SP960, which can be used for the sequencing of the library preparation automation for 96 samples in one run. So it can complete within 8 hours. So apart from the sequencers, we have our software called ATVOplex, so which can automate and which can multiplex the alleles. For example, if you need to prepare any panel by multiplexing of your alleles, for example, STR alleles, so then we can use this ATVOplex software, which can multiplex up to 20,000 of the alleles into one run, one tube. So that means you don't need to prepare for 20,000 of the STR alleles, you don't need to prepare 20,000 PCRs. All the 20,000 alleles can be multiplexed into one tube only. So that means one PCR for only one sample, up to 20,000 alleles. So especially this ATOplex software will be highly useful for the data analysis and also library preparation of wildlife samples, especially for the non-invasive samples. For example, wildlife non-invasive samples will be dung samples, or maybe especially for the environmental DNA samples, also water and soil samples. So because the quantity of the DNA will be very, very low from this one. So the panel that is designed by ATOplex platform that can be used for quantity of the DNA down to 0.1 nanogram. You don't require even 1 nanogram of DNA. So this ATOplex that can be used for the multiplexing of the PCR alleles for up to 20,000 samples, and it can be easy to run, and also it is very low expensive compared to the whole genome sequencing. So here we are providing all the consumables starting from the extraction to the library preparation to using the panel designed by ATOplex platform, followed by the sequencing and data analysis by Megaboard. And also if you need to integrate any AI tools or machine learning tools, that can be integrated into the Megabolt also for the data analysis also. So this can be, ATOplex can be used for the panel designing of DNA libraries or RNA libraries also. So already we have prepared multiple ready-to-use kits, especially for the environmental DNA samples, for example, 16S for the bacteria, and also ITS for the fungi, and ATS for the nematodes and proteids. And apart from that, recently we have customized one panel for WIA for the 11 STR tools, STR alleles for the elephant samples. So what does it mean by ATOplex? A stands for the auto workflow. So that means the panel designed by ATOplex that can be prepared, that can be used for the automation instruments. Okay, now you don't need to use